have some jumping in now. All right, good afternoon and welcome to today's webinar, Harnessing Genetic Information for Better Mental Health, brought to you by the University of Iowa, Division of Child and Community Health, Child Health Specialty Clinics, and the Regional Autism Assistance Program. I'm Shawn Wittrock and I will be moderating today's session. The session will be recorded and the link to that recording will be provided in a follow-up email. You are free to type your questions into the question box throughout the presentation and those will be answered at the end. CE credits will be available for those attending this live webinar, and directions for obtaining those credits will be shared at the conclusion of the presentation. I am pleased to introduce and welcome our presenter, Dr. Jake Michaelson. Dr. Michaelson is a Roy J. Carver Associate Professor in Psychiatry and Neuroscience and the Division Director of Computational and Molecular Psychiatry at the University of Iowa. His lab studies the effect of genetic variations on the development of the brain with specific applications in autism and language impairment. He earned his Bachelor of Science and Master of Science in Biological Engineering at Utah State University before earning his PhD in Computational Biology at the University of Dresden in Germany in 2010. After his time in Germany, Dr. Michaelson joined the lab of psychiatric geneticist John Sabat at UC San Diego, where he completed his postdoctoral training and published several of the earliest papers dealing with whole genome sequencing in autism. In 2013, he joined the faculty at the University of Iowa, and his current research is supported by the National Institute of Mental Health, the National Institute on Deafness and Other Communication Disorders, the Simons Foundation, and the Brain and Behavior Research Foundation. His research areas include genetics and comorbidities of autism, genetics of language impairment, predictions of top autism gene candidates, and genetics of twice exceptionality. Without further delay, I will turn the presentation over to Dr. Michaelson.
Great, thanks for that introduction, and I'm grateful to be here with you guys to tell a little bit about some of the research that we've been doing. And I just want to let everybody know that, so I have no disclosures. I recently gave a very similar talk to this in my department's grand rounds here in the Department of Psychiatry. I think that some of the material might be a little bit technical, and so I'm going to breeze through that. Certainly, if you have questions about it, I'm happy to field those, but just in the interest of time, I may gloss over some of the more technical aspects. So here's the title slide for my talk, and I'm kind of a tarot enthusiast, and so that's why I put these cards on there. I think it's a very interesting. Now I want to be clear, as a scientist, I don't put any stock in terms of, like, you know, the science behind fortune telling or anything like that, but I do think that it is an interesting way to motivate thinking about yourself and about your strength and risks that one might be facing. And so I just wanted to put this is New Year's Eve at our house, and we do a tarot reading that has evolved into much more complicated things, but the kids love it and they ask for it every year, and it's kind of a fun opportunity for us to have kind of a conversation that thinks about what strengths they have and that they can use in the coming year. And so, like I said, this is a sort of a side hobby of mine, and I think it's interesting in the way that it provides a tool for introspection, and it can help you think about what the challenges that you're facing are and what your strengths are and how you can bring those to bear on things that you're facing in your life. Now, in a much more objective and rigorous way and in a scientific way, I think that knowledge of one's genetic information can likewise help us to consider in a new light the connection between resilience and risk and well-being. And sort of fitting on this theme, I had a conversation with my sister yesterday that I thought was appropriate for some of the things I was going to be talking about today. And she forwarded me this link to this company that will take your money, and if you give them a sample of your hair, they promise to return test results back for 970 items of things that you are particularly sensitive to. And she wanted to know, like, is this legit? So I had a quick look at the website of what they had to say about their methods. And, you know, I read this, and immediately it was clear to me that there was no actual science behind it. In fact, I don't think that they are really even doing any kind of testing to the sample that they send you. I think they're probably just taking money and then giving you what is essentially a random report. It's basically just technobabble. But the lesson for me in this was that I think it's interesting that the fact that people are willing to pay for this, it speaks to this innate craving that we have for medicine to be more personalized than it currently is. People are very interested in, and they have this innate sense that, I do have an individual biology and an individual physiology, and my individual variations have some kind of a role to play in how I respond to treatment. And so I think even though in this case there's certainly no science behind this product, I think it does speak to people's desires for it to be for medicine to be more personalized. So what I'm going to talk about today, this will come back later because I'm going to talk about some of the ways that we can use actual science to acknowledge the individual level diversity and how people might respond differently to different medications, for instance. So I'll spend a little bit of time giving some basic background in genetics because I'm going to talk about, how these genetic variations factor into a couple of different applications in psychiatry. I'm going to take a brief detour into a couple of slides about genetics and social justice because I think that's an important emerging point of discussion. And then the two application areas that I want to focus most of the time on are matching medication to personal biology, and then using genetics to maybe assist in the diagnostic process, and especially in things like differential diagnosis. So this is representative of a stretch of DNA. So all living organisms have DNA. Humans have about 3.2 billion letters of DNA. So these individual we call them nucleotides, make up the DNA code. And so we have a lot of this DNA sequence, and there are specific places in the genome where we know that there is genetic diversity. In other words, in this example here, we know that it's not uncommon for a person to have a G in this particular position. It's not uncommon for them to have an A in this position. And so we call versions of DNA strings, or you could think of them as genes, we call those alleles. And so we would say that this is the reference allele because that's the version that's in the human reference genome. And then any variation from that reference we consider to be an alternate allele. And this is what you may have heard people talk about SNPs before. These are single nucleotide polymorphisms. So it's just this position right here where we know that there are differences in the letters that people have. And because we have two copies of every stretch of DNA, one that we inherit from our mother and one that we inherit from our father, there are these possible genotype states. So these different combinations of alleles that we can have at different positions in the genome. So here in this example, we have a homozygous alternate, 2 Gs. We have heterozygous, which is one version that's a G and one version that's an A, and homozygous reference. So this is just to key you into the terminology here. When I talk about heterozygous, I mean that you have one of each, and homozygous means that you're both the same at that particular position. So probably the most important thing is just these are SNPs. These are single nucleotide polymorphisms, changes or differences in the genome that we know to exist. Now, a lot of times we want to connect genetic variation to variation in a trait. And here I'm just using as an example, one of the things that we study in my lab is language ability. And that's something that we can assign a quantitative score to as a trait that we want to understand in genetic terms. And so we may look at some positions in the genome, and we can group people's language score. We can group people and their language score according to what genotype these people have at a particular place in the genome. So this is just looking at one place in the genome, and we have some people that have CC, some people that have CT, and others that have TT. And you can see that among those three groups, there's really no difference in the language score that they have. And so this is a non-associated SNP or polymorphism in the genome. This has no bearing on your language ability. Now, in contrast, there might be some other place in the genome where we could group people similarly by the genotype they have at that position in the genome. So here, some people have AA, others are they're heterozygous and they have AG, and some have GG. And in this instance, we see that there is a relationship between what genotype you have at a particular position and your language score, right? So we would say this position in the genome has some kind of influence on your language ability, and we would like to understand more about the context around there. What gene is that in, and what do we know about that gene? Is it involved in brain development? This is something that geneticists do to help us understand the relationship between genes and traits that we're interested in. So if you were to do a similar kind of test, like with these box plots that I was showing you, if you were to step through the genome and look at hundreds of thousands or even millions of different places in the genome and do that test for every one of those places to see if there's a significant relationship between the genotype group and the trait, you might end up with something like this. We call this a Manhattan plot because it looks like the skyline of a city. And the peaks that you see are simply places in the genome where we see a very statistically significant association between the genotype group and the trait value. So it's just here it's expressed as the negative log 10 p-value of that association. So higher means a stronger association. Now, for most places in the genome, there's not going to be any genetic association between your trait and what the genotype group is at a particular place in the genome. And so that's what you can imagine happening at these places where there is no peak. But for places where there is a peak, you can imagine that there's something like this happening where what version of the gene you have matters for the value of the trait, in this case, this language score. So the genes that correspond to where these peaks are genes that we would want to study further in the context of language in this example. So this is what we call a genome-wide association study. And again, this is kind of a part of your typical gene finding exercise. But another really interesting thing that we can do with once a GWAS is performed, this genome-wide association study, is we can take what are called the summary statistics, which is essentially the effect that genotype has on the trait. Could be a positive effect, meaning that it increases the propensity of that trait. Could be a negative effect, meaning that it reduces the propensity towards that trait. And we can aggregate all of those from all across the whole genome, and we can distill it down into a single score, just as a simple linear combination of those effects. And at the end of the day, we get a single score that says, according to all of the variation that we observe in your genome, this is your genetic propensity for this trait. It might be for schizophrenia, it might be for autism, could be for educational attainment or your height. Right. So, but we can develop these polygenic scores that are in large samples predictive of those traits. And we use those in research applications. But I'm going to talk a little bit about how these might be used in the near future in personalized medicine. So I'm going to pause briefly and talk about what is heritability. This is a word that you might have heard before. It's a measure that its value is between 0 and 1. Can't be higher than 1, can't be less than 0. And in essence, the only thing you really need to pay attention to are maybe these first two bullet points that I'm talking about here. So in practical terms, if you consider some trait X, like this language trait that I was talking about, it's heritable if, in the case of you inheriting different genes, you can expect your life outcome for that trait to be different. So if you inherited a different set of genes and you would expect your language ability to be different, well, then language is heritable. Another way to look at it is that heritability is asking how much of the differences in trait X, or something like language, for instance, or height, are explained by genetic factors. So this is similar. You may have heard of an R squared before, which similarly is bound between 0 and 1. But this expresses how much of the variation has been explained by your predictor variables, which in this case are the genetic factors. A couple of other interesting things I just want to point out is that heritability is not necessarily constant. It's something that can change over time. It can change between populations. It's not like, a universal, like a gravitational constant or something that we expect to be the same everywhere, but it is subject to various influences. It's also a population parameter. That means that it's specific to the population in which it was measured. So just to give you a few examples of the range of heritability and to develop some intuition here, some of the traits that have lower heritability are many cancers, not all cancers, but some cancers have fairly low heritability. which is kind of interesting because cancer still has a lot of genetic influence, but it's not necessarily heritable. A lot of that can arise from mutation, mutations that happen in individual cells within your body. Things like handedness, that has relatively low heritability, although we've done genome-wide association studies on handedness, and there is some genetic signal there, but it's relatively low. Things like political beliefs. There are certain personality traits that are more highly heritable than the political beliefs that are sometimes associated with them, but your political beliefs are often heavily influenced by your environment and by the way that you were raised, for instance. But some higher heritability traits include things like height, very highly heritable, eye and hair color, those are very heritable. Psychiatric disorders like schizophrenia, bipolar disorder, neurodevelopmental conditions like autism and ADHD, those are highly heritable. And so why does heritability matter? Well, it's one of the first things that we look at as geneticists to determine whether or not it's worth investing effort in continuing genetic research for a specific trait, because if there's a trait that has very low heritability, then it's probably not going to pay off to invest a lot of time and effort into genetic research. It can tell us a little bit about how malleable or how plastic or how modifiable a trait can be. It can tell us a little bit about whether interventions should target more of the embodied factors that are related to the trait, or maybe the more circumstantial or environmental factors that are associated with the trait. It can also, I'm going to tell you in a few slides, it can influence the discussion about how greater equality and equity and justice can be attained from a policy perspective. So one example of influences over heritability, I think, is a really good one for women, right? Because women in the recent past have faced a lot of barriers and a lot of oppression in terms of advancement, specifically around education. And so you can imagine that perhaps for men, educational attainment might have been more genetic or more heritable than it would be for women, because women have these artificial constraints put on them. And we see that over time, as there are fewer and fewer barriers to women achieving higher educational attainment, that their ultimate educational attainment becomes more and more genetic. The heritability goes up as those barriers are removed. Other traits, things that intersect with cultural and societal norms, things like sexual orientation and gender diversity. So we expect, as further studies are done on these things, as stigma comes down and as persecution comes down, we expect to see an increase in heritability of these traits. So, and then these are just kind of some of the other things that influence our estimation of heritability. And so I'm going to spend a little bit of time talking about what genetics are able to predict in terms of psychiatric diagnoses. But before we get to that, I think it's worth kind of calibrating our expectations. What proportion of variance is explained, thinking about the R squared of some pretty common everyday intuitions. So a lot of us have allergies, we take antihistamines for them, but the relief that we experience, there's very low predictive power in getting relief from your allergy symptoms and taking an antihistamine. Things like that men weigh more than women, that's only a 7% R squared. Only 7% of that variability is explained. Places at higher elevation or colder, 12%, 19% for the taller people weigh more, 11% that wealthier people complete college. So one of the points that I just want to make here is that for heritable social science outcomes, like educational attainment is a big one, the attempts to predict, even using non-genetic variables, but using every variable that you could possibly imagine at your disposal, they would rarely exceed explaining about 20% of the variation in that trait. And so that's kind of the ceiling of what we would expect, and that's about where the bar that should calibrate our expectations when we think about how much genetics is predicting. So I'll give you a concrete example. Some of the largest genome-wide association studies have been on traits like educational attainment, because that's a fairly easy thing to measure over very, very large samples of people. And so you do a genome-wide association study, which is like that Manhattan plot that I showed you before. You identify places in the genome that where the variation in the genes is associated with educational outcomes, educational attainment. And then you can compute in a totally independent sample, you can compute scores based on a person's genotype of what you expect their ultimate educational attainment to be, and you can see how well that predicts. So in general, the R squared for a polygenic score for educational attainment, that's about 15%. And remember, I just told you that 20% is about the absolute highest that you would expect, even not even using genetic data. But to put this in real terms, if you divided this up into quintiles, this score, in the bottom quintile of the polygenic score for educational attainment, there's only a college completion rate of about 10%. That's this right here. But in the top quintile of polygenic educational attainment, college completion is 60%. Right. So I just want to emphasize that these polygenic scores, if the genome-wide association study that it's based on is well powered, has a large number of people, and the heritability is sufficient, you can really predict quite a bit about what is in store for a person in meaningful life outcomes. So similarly, in the genetics of height, there was recently a GWAS that was published where they reached saturation, meaning that they had enough people, they had 5.4 million participants in this study, and they basically reached saturation in terms of what could be explained by common genetic variation, the kinds of genetic variants that they were assaying here. And they found 12,000 independent places in the genome that are exerting an influence, positive or negative, on human height. And 45% of the variation in height is explained by these factors. And just to kind of make this concrete, I computed the polygenic score for height for me, and it was within an inch of my actual height. And you could chalk the rest of that up to environmental factors. Maybe, you know, if I had had a different diet, maybe I would have been taller, maybe I would have been shorter, you know, if I had other different upbringing or something like that. But it's very predictive. So now for psychiatry, though, we still have a little ways to go, although we are starting to get really meaningful results. The latest GWAS on schizophrenia, which is the largest GWAS among psychiatric conditions, had 75,000 cases and about 100,000 controls. They identified 287 different places in the genome that are significantly associated with risk for schizophrenia. And this paper was published in 2022. There's already been 246 other papers that have cited this paper, and many of them are following up. They're pursuing mechanistic experimental studies of the candidate genes here so that we can start to think about what the mechanisms are and potential therapeutic approaches. So this was kind of aimed a little bit more to the providers, but might have relevance to some people on the call here. But one of the things that you can do is, as a provider, that is recommended, if you're working with anybody who has any kind of a developmental disorder, then the recommendation is to order a microarray. And the purpose for that right now is to look for very large, what are called copy number variants, deletions and duplications that could explain that person's condition. So there, you know, I think in general, it's a good idea to order this if it's available, and especially if insurance is going to cover it. So again, here are the cases where it's very routinely ordered in the cases of intellectual disability, autism, language disorder, ADHD. We even had some people with adult onset psychiatric conditions that have successfully had insurance pay for a clinical microarray. So just to recap this part, heritability is a measure between 0 and 1 that gives us a sense of how genetic a trait is. Psychiatric traits are highly heritable. Autism is above 80% heritable. We don't really talk about the predictive power of the R squared, but unlike height, psychiatric genetics has not yet reached saturation, but it's already changing our understanding of these conditions and providing new therapeutic leads. right? So we're nowhere near the million. So this GWAS was 31 times larger than the schizophrenia GWAS, which is the largest genetic study in psychiatric conditions. Okay, I'm going to take a brief detour and spend just a couple minutes talking about genetics and social justice. And this is largely based on a book that we read in my lab we have a lab book club that we rotate through different books that are written for a lay audience, but that definitely have a scientific foundation. This is from Catherine Page Harden, who is on the faculty at, I think it's UT Austin. And she does genetic research in social traits. And basically her core argument in this book is that if you imagine the ideological spectrum, there's abuse of genetics on both ends of that ideological spectrum, but the abuse is in very different ways. So on the right, we have the typical argument that genetics imply and potentially even justify a natural social order. And that leads to things like eugenics and racism. And then on the left side, we often see attitudes like, well, there are no genetic differences. And if there are, they're not meaningful. And that can lead to other problems that are related to the very real role of genetic factors in many of the ways that our current world is not just. And so she kind of develops what she calls an anti-eugenics approach that basically acknowledges that there are meaningful genetic differences between individuals, and those differences do drive meaningful life outcomes. And she advocates for the position that society should be structured as a prosthetic, and I'll get to what I mean by that in just a second, that decouples the genetic lottery. And she calls it a genetic lottery because nobody did anything to deserve the genes that they inherited. They, in effect, were not even really around when their genome was established, and they played no role in deciding what that was going to be. So it really is a lottery. But to decouple that genetic lottery from health, education, and happiness. So an analogy is kind of like with glasses. So we, I think we can use this as a way to understand how we can divorce this idea of basic human worth from the genetic lottery. So poor vision, that's undeniably biological in nature. And nobody denies that there are innate differences in vision. It's not seen as a character failing. It's easily corrected with the prosthetic, and in this case, that's glasses. And it restores functional parity with non-impaired peers. And if you do this thought exercise, imagine doing a genome-wide association study on corrected vision. You'd come up empty with that. You wouldn't find any genes that are associated with vision if you have a prosthetic in place that corrects vision to the level of your peers. So you'd have heritability of 0 for corrected vision. Another way to think about this is to, if you, this is again a thought experiment, but imagine a world where there was a very repressive environment, and there was, some constraint placed on women that women had to wear broken glasses. And obviously, your vision's not going to be great in that. And so in a repressive environment, you see low heritability because there are these artificial constraints put on people. Once you reach equal opportunity, where you've kind of leveled the playing field, then you start to see high heritability of traits. And then as you move into more of a societal prosthetic zone, then you again can see a reduction in the heritability of a positive life outcome because you are putting things in place to kind of make it so that everybody is able to achieve happiness and fulfillment regardless of what their genetic potential is. Okay, so this is kind of just a general schematic of the relationship between how heritable or how genetically determined something is and our societal progress towards leveling the playing field so that everybody has the opportunity to be happy and fulfilled. So that's just my digression there, but I wanted to give you a window into our thinking as we went through this in our book club, and also for the remaining things that I'm going to talk about for the rest of the time here. So This part I'm going to talk about matching drugs to personal biology. So right now, the system that we have to match drugs to their indications, it's based on clinical trials. And those have to be very well controlled, and they're very hypothesis driven. And so what this approach makes a couple of assumptions that are really important when you think about how slow things move on this front. So first, it kind of assumes that we have a comprehensive understanding of the disease mechanisms, which certainly is not the case. There's also this implicit assumption that there's relatively little disease heterogeneity, meaning that the cause of the disease is relatively singular. And then there's also a built-in assumption that there's going to be a uniform response across different genetic backgrounds, different ancestries, which we also know to not be the case. But there's a complementary approach, and I'm not saying that this should completely replace the clinical trial approach, because I think that's been pretty important in ensuring the safety of a lot of these approaches. But I'm going to describe what's called transcriptome-based drug repositioning. And this makes very few assumptions. The only thing that it assumes is that if you are able to reverse some disease-associated gene expression signature, and I'll get to what that is in just a minute, if you're able to reverse that gene expression signature that's associated with a disease, then that implies that that drug would be effective against that disease. And another key thing is that we can impute, or we can estimate, gene expression from the kind of genotype data that I just described before with these SNPs and the kind of genotype data that's used in genome-wide association studies. It's also the kind of genotype data that you may have gotten for yourself from 23andMe or Ancestry.com or other direct-to-consumer genetics companies. But these can be used to identify new drug applications and prioritize drugs for optimal individualized response. And so I get a lot of questions about epigenetics, I think largely because it's a buzzword that people have heard in the popular media and stuff like that. But really what epigenetics is all about is how an individual genome is expressed. It's all about gene expression, because the act of gene expression leaves behind these marks of varying durability. And so DNA methylation is an example of one of these. It's probably the most popular. It's usually what people are talking about when they talk about epigenetics. But there are also other epigenetic marks like histone acetylation and so forth. And these marks they may be a proxy for exposures or experiences, but ultimately it's about the gene expression that happens in response to those things. And so when a provider prescribes a drug or an intervention, what they're really prescribing is some kind of epigenetic reprogramming, because that drug is going to induce a change in gene expression that is going to change the character and the responsiveness of certain kinds of cells. And we hope that that reprogramming will have therapeutic effects. So again, the general idea behind this gene expression-based approach is that each of us expresses the same genes in different ways, right? So here I'm just showing 3 different people, and this is just a sampling of some of the genes in our genome. We have over 20,000 different genes, but this is actual data, and it just shows you that some people will express a certain gene more highly than other people at baseline, and some people will express some genes lower than others. Some of that is just benign variability that just contributes to our individuality, but some of that variation might explain symptoms. It might explain complaints or disorders that we have. And so the core premise is that your individual gene expression signature, so when I say signature, I mean kind of like this, the ups and downs, if you were to look over the entire expressed genome, your personal ups and downs there, and specifically that perturbation from the mean or the population mean, that provides mechanistic clues about your personal biology and your personal pathology. So I'll give you an example here from a study that we published a couple of years ago. In 2015, Alex Basuk, who's now the chair in pediatrics, approached me about collaborating on a gene expression study of brain tissue from neurosurgical samples. These are people who have treatment refractory epilepsy. It's very difficult they tried, essentially every other option and their last resort to control the seizures, which are debilitating, is to have a piece of their brain removed. And so it's a very, it's kind of the last resort. So this started off as a typical differential expression study. And what that means is that we just look at two conditions and we want to just check to see what genes are expressed differently in those two conditions. And so for this, we looked at spiking tissue, or tissue that was experiencing seizures, versus the neighboring tissue that was not spiking. It was not experiencing seizures. And we just looked at what the gene expression differences were between those two groups. And we got a list of genes. We looked at it and we said, okay, this makes sense for what we know to be happening in terms of seizures. But we wanted to get more out of this. We didn't want to just stop there. And so around the same time, there was this new approach catching on where other scientists were doing things like they would grow a bunch of cells in a dish, and then they would expose those cells. And, you can imagine on a plate like this, they would expose those cells to a vast library of compounds or drugs. And then they would simply measure the resulting gene expression that was perturbed by the introduction of these different kinds of drugs. And so just like you and I have our own gene expression signatures, these drugs each have their own gene expression signature that they induce as a result of them kind of perturbing the cells. And so we wondered what drugs would show the opposite pattern, right? Because we discovered this pattern that was related to epilepsy, the spiking versus non-spiking gene expression pattern, and that gave us a certain pattern. We wanted to know what drugs produced the opposite pattern to what we were observing in seizures. Because in theory, if we expose the diseased tissue to a drug known to invert that problematic pattern, that drug might be therapeutic. So here's just kind of a rough diagram of how we went about doing that. But in the end, we validated, we came up with a list of drugs. We validated them in a zebrafish model, which I think, I have on this slide here. There's a model of seizures in zebrafish, and we took a couple of these compounds, and we showed that it basically eliminated the seizures in these zebrafish. Right. So we thought, okay, that's really cool. We stopped well short of trying any of these out in humans, but these were largely drugs that were already approved by the FDA, but nobody had ever thought about them in the context of seizures before. And so we published that paper and we felt pretty good about it. But fast forward to this past year, a French-Canadian mom came to us and she told us the story about her 8-year-old son who had intractable seizures since age 2. They tried everything. The neurologists were stumped. She had another son that was diagnosed with roundworm, and so the whole family began treatment with Parentel, which is, as you can see in this cute little diagram here, is an anti-worm agent that the whole family began taking. Once they started taking it, they noticed that the 8-year-old seizures stopped, which was incredible because nothing else that they had tried. They tried all sorts of medications and nothing worked. And so there were no seizures for 2 weeks after he took Parentel, and then they ran out of the medication. Since then, she's rationed what little she's been able to get a hold of because the providers are dubious and they're reluctant to prescribe. So she came to us because they found that it was our paper that recommended this exact drug as a potential treatment for treatment-refractory epilepsy. And in this case, it was a very serendipitous human in the wild kind of a trial. It worked. And so I think this is worth pursuing. And at this point, based both on our research and this remarkable case report, I'm a believer in this approach of identifying gene expression patterns that are related to disease and then trying to find drugs that reverse that pattern. So we can't measure gene expression directly in everybody, but DNA genotype, like I described before, like 23andMe has, that's fairly plentiful. And I'm going to kind of, just in the interest of time, I'm going to kind of gloss over this. We can come back if there are questions about it. But again, the idea here is that we can, just like we can compute a polygenic score for educational attainment or for height, we can also compute a polygenic score for an individual's gene expression, right? So we can go gene by gene and estimate, based on this person's genetic makeup, do we expect them to be a high expressor of this gene or a low expressor of that gene? And that can help to get us a gene expression pattern. So I'm just showing here that this is you can go to your 23andMe account, you can download the raw data. And I did that for myself. And we applied our pipeline, our methods to it. And then from that genotype data, we have estimates over these different brain regions of which genes I tend to express more highly and which genes I would tend to express at a lower level based on my genetics, okay? So this is an important foundation, because what we're going to do next is we're going to try and we're going to look through a whole huge library of drugs to find drugs that would be the opposite of what my gene expression pattern is, or you could imagine what a patient's gene expression pattern is. So again, this is an individual's gene expression signature that we imputed using the genotype. If we looked at the average pattern across many people, for instance, if they were diagnosed with ADHD, and we might see a pattern like this, that there are some genes that we see more highly expressed on average and some that we see more lowly expressed. And then this is the pattern induced by drug exposure. So here, methylphenidate or Ritalin is something that's used for the treatment of ADHD. And so you would try to see how well that matches up or reverses this pattern, right? So for instance, you can see here that people with ADHD tend to express this gene highly. And if you have methylphenidate, it drives down the expression of that gene. So that would be the kind of thing that we're looking for. So here's another couple of examples of where that pattern is reversed. Right. So I'm going to, again, kind of gloss over some of the more technical details here, but the basic idea is that we are estimating each individual's gene expression, not once, but in every different kind of tissue that they have, because as we know, different diseases have kind of different tissue relevance, different organ relevance. We're most interested in the brain, and so we impute gene expression that relates to the brain. And so we can do some sanity checks, like, for instance, if we predict that some people are going to respond to a particular antidepressant better than others, are the people that we're predicting as responders, are they more likely to experience depressive symptoms just in general? And likewise, are people that we are predicting to be stimulant responders, are they more likely to exhibit attention symptoms? And then finally, do predicted responders improve symptoms upon medication more so than non-responders? So I want to make an aside here. You've probably heard of pharmacogenetics before. This is not that. Pharmacogenetics typically genotypes just a small handful of SNPs. As a field, it's generally much more focused on the drug kinetics, and it's focused on genes called cytochromes. It's not terribly tissue specific, and it is commercially available. This is something that's not commercially available right now. It's genome wide, it's focused on mechanisms, and it's tissue specific. So our first sanity check was that we looked over 523 drugs that are commonly prescribed in psychiatric applications. And then we calculated for each of those the drug level, what we call polygenic responsiveness to each of these 523 compounds. And we grouped them according to the mechanism of action, and we correlated that with individual measures of problem behavior from the child behavior checklist. And so this gives us a map of mechanism to behavior, to the behavior that it's expected to ameliorate. And so this is just a heat map. And all I want to point out right here is that if you these are the behavior scales in the rows here, and then these are the different drug classes in the columns. And if you look at a few places like the attention deficit hyperactivity, and the peak match is basically with dopamine reuptake inhibitors, and this is one of the mechanisms by which stimulants act, right? So rule-breaking behavior, we also see traces of dopamine. Social problems and being withdrawn and depressed, we see SSRIs being something that is expected to be effective, right? So this is kind of our sanity check on that. We also did a further test looking in a cohort that had multiple time points, and had medication data and also had behavioral outcomes. So we looked specifically at methylphenidate, and we wanted to know, based on people that we predict to respond to methylphenidate and those who don't, who we predict do not, do we see any differences in how they respond, right? So in the blue are the positive responders in year one. as they are on methylphenidate by year 2, here's year 0, here's year 1. So this is like year 1 and year 2. Their attention symptoms have gone down in the positive responders. In our group that we predict to be negative responders to methylphenidate, the symptoms actually go up a little bit. And this is something that we see as we kind of slice the data in different ways. So if you discontinue methylphenidate, we see kind of no change year over year in the people who are responders. But if you're a negative responder, your attention symptoms actually improve by going off of methylphenidate. In the interest of time, I'm going to kind of skip over that. I think I've got about 5 more minutes of material here, and then we'll get to questions. So this is just to sum up here, we have developed, and others are working on similar methods, but we've developed basically a way to take genotype information and make predictions about how you express genes at a steady state at baseline. And then we can make predictions about what drugs might be most appropriate to you based both on obviously your symptoms, but also on your own personal physiology and how which genes you express at a high level and which at a low level, and match genes in a more match drugs in a more individualized way. Okay, for the last just couple minutes here, I'm going to talk about applications in differential diagnosis. So family history is a really key part of a psychiatric diagnosis. It's quick, it's free, it has very robust information content, but it has some downsides. So it doesn't give us really any mechanistic insight into what is actually causing these problems. The other thing is that a good family history depends on family size and intra-family openness and the level of stigma, right? So if you come from a very small family, then family history is of very limited usefulness. And even if you came from a big family, if nobody ever talks about things that they're going through and you have very little insight into what your family history actually is, then it can be less useful there too. And so this is where genetics, and specifically these polygenic scores, I think can be very helpful. And so I'm just going to present 3 cases that are actual, real cases of people who are in both our registry, but who are also patients at UIHC. And we've calculated polygenic scores for the main psychiatric conditions. And I have a picture next to each one of them, but I want to be clear that, before I get to that, right. So for each case, consider what, if anything, and this was geared towards providers, would change in your handling of the patient if you knew about these genetic scores. How, if at all, should these be communicated to the patient? And how would you feel if you were in the patient's shoes? So this person does not exist. This is kind of an interesting website that can generate realistic looking photographs of people that aren't real. But I thought it would be helpful to have a picture just to kind of communicate this is a real person, but this is not what they look like. So here we have a 41-year-old female who's been diagnosed with major depression, also with generalized anxiety disorder. But in this case, the polygenic risk, if we look at where there's risk, there seems to be a lot more going on than just these diagnoses. So the highest level is for ADHD, also very high for autism, very high for bipolar, and also high for schizophrenia. But the thing that she's been diagnosed with, major depression, she has this is like low average polygenic risk. And so her condition is actually much more complex than what these diagnoses would reflect. So that's case 1. Case 2 is a 45-year-old female diagnosed with bulimia, with bipolar disorder, with PTSD and IBS. She has very high risk in the 99th percentile for autism, and also high risk for cannabis use disorder and elevated risk for schizophrenia. But she's been diagnosed with bipolar, but actually has very low genetic risk for bipolar, also very low risk for ADHD. And so the fact that her highest level of risk comes from autism, but these are the diagnoses that she ends up with, I think is something worth thinking about. And then finally, this case 3, I think is interesting. This is a 10-year-old male. The diagnosis was ADHD with an expressive language disorder. Polygenic scores for ADHD and autism were both well within the very normal range. But what stood out was that this young man has a bipolar 1 risk in the 99th percentile. Now, the thing that's, I think, important here is, although adolescent and pediatric onset bipolar is definitely a thing that people understand happens, it's not necessarily the first thing you're thinking about as a child psychiatrist. You're probably more used to things like ADHD, and if those symptoms match, then that would be the diagnosis that makes sense. But really, the underlying risk is very different than ADHD, and this person should probably be monitored and followed up with in a way that's different than a typical ADHD case. So just to sum up, these are not, I think very soon they're going to start to enter, in the next couple of years, we'll start to see these more in the more pioneering clinics. It's not going to replace the current diagnostic process, but I think it could make providers think twice as they're considering the different possible diagnoses that might fit. And it can be especially helpful when there's a very scant family history, or if it's noisy. Yeah, so these are the take-home messages, and just in the interest of time, I'll stop right there and we can go to questions.
All right, thank you, Dr. Michaelson. Like you said, we do have some time to take questions, so please type your questions in the Q&A section. And while we wait for questions to come in, I will follow up with a few housekeeping items. So for attendees who would like to claim continuing education credits, the UI Carver College of Medicine has an online portal to complete the course evaluation and download the attendance certificate. And you will receive an email with step-by-step instructions on how to access that online portal and complete the evaluation following the webinar. So I will also put that link to the evaluation in the chat now. And if you do have questions regarding continuing education, please feel free to contact me, and I will also put my contact information in the chat box. So far, no questions. So I don't know if you wanted to kind of go through these take-home messages a little bit more. Or...
Sure. Yeah, so just to overall, the whole presentation, that these polygenic scores, hopefully now you at least have a sense of what they are, that they're kind of distilling down your entire genome into a single number that kind of estimates your propensity for a certain trait. So you'll start to hear more about these, I'm sure, in the mainstream media in the next couple of years. But they can predict a lot of interesting things like height, educational attainment, and risk for mental illness, just to name a few. So I think these will become clinically useful probably within the next 5 or so years. And I think they really do stand a chance of really individualizing the treatment a little bit more. And you know, when the symptoms might be a little bit ambiguous, it will help in terms of the differential diagnosis to point towards better informed treatment options. And then the other thing that's sort of related to computing these polygenic scores is that we can also, in much the same way, impute or estimate gene expression, because that's really what, you know, a big part of determining our individual biology, our individual physiology, is how our genes are expressed at a steady state. And we've used this as a means to gain insight into what drugs will stand the best chance of reversing or normalizing disease-associated gene expression patterns.
We did have one question come in. How often is a genetic panel available for someone with a co-occurring psychiatric condition and substance abuse?
How often is it available? I guess I'm not sure what how often means, but because actually the nice thing about the genetic panel is you only really need to do it once in your life, because your genes aren't changing in any kind of way that's meaningful. But with a co-occurring, so for adults, I guess is sort of what you're asking, having a psychiatric condition and substance abuse, maybe young adults, that is a little bit more open question about whether there would be reimbursement for it. Certainly it could be ordered in any case, but you know, it might change how much you have to. And I think I should not be quoted on this. I wish Ben Darbro was here to tell me, but if a person ends up having to pay out of pocket for it, I think it's probably in the realm of like several $100s or so. So I think that's probably the limit on how expensive it would be if it were not reimbursed by your insurance.
No follow-up to that, so must have answered the question. Who is eligible to participate in the registry and complete the studies for the polygenic scores?
Yeah, that's a great question. We do have a study that we are that's open enrollment right now. I wish I had a slide up describing it. You may have, if you're on the general mailing list for the University of Iowa, then you probably would have seen one that was asking about whether you're a high achiever and to participate in neuroscience research. That's something that we're doing right now that's kind of part of our twice exceptional stuff. But actually anybody with a neurodevelopmental or psychiatric condition can join our registry. Unfortunately, I think this is something that will have to change soon. IRB and others are kind of skittish about the return of genetic results to people. And so in the past, we haven't been really forthcoming about returning those results to people, but we're also developing tools that people can do it themselves. So like, for instance, if you have 23andMe data, we are developing pipelines that people can upload their own data and compute some of these scores. I realize this is kind of a meandering answer to your question, and I wish I had a really direct. I wish I could say we are actively doing this study enrolling people into this study, because everything I've shown you is based on data from people who've been in registries for a long time. But this is kind of the frontier that we expect within the next year or so to be. We're going to have a new registry that is for neurodiversity in general, so it include anybody with any kind of diagnosis, psychiatric diagnosis, or any other indicator of neurodiversity. And that one is going to be heavily polygenic oriented. So I guess probably the best answer I can give is to stay tuned and keep your eye on your email inbox if you're on the listserv for the University of Iowa. And you can always email me too, and I can send you details about how to join the registries that we do have open right now.
Nice. Thanks so much. Very interesting. Thanks for the book tip on the genetic lottery.
Great. Yeah, we really enjoyed it.
And thank you for a stimulating presentation.
Yeah. Thanks everybody for your questions.
One
John,
more
this is a question. So patients with intellectual disability has microarray done, results normal. What does that mean? What that means right now is that they looked for a specific kind of fairly large genetic variation, and they didn't find it. That doesn't mean that there's not genetic factors. We're actually very interested in these people who do have a microarray done and there are no findings, because what we think we can look for are these polygenic scores, right? So because it's not part of routine clinical practice right now to compute these polygenic scores and then return anything that might be out of the norm. But we do think that in many of these cases that might be a contributing factor. So that would be another thing that we're actively thinking about right now. And we're working with Ben Darbro on a project where we're going to be doing that. Now, how long it's going to take before that is made a clinically routine thing, I don't know. But I suspect that people who enroll in the research part of it, part of that will be that you'll get results back with some kind of a guide to interpret them.
All right, well, it doesn't look like we have any additional questions that have come in. So this will conclude our presentation for the afternoon. I want to sincerely thank you, Dr. Michaelson, for presenting, and for all of you who attended. Have a wonderful afternoon.
Thanks, everybody. Bye.